Hi Year10s, welcome to our week 2 video on cell division. So this is a new one added for 2023. So the order is probably a little bit different than what was on the website originally, but this is now a new video, and there'll be 3 videos in this collection, all on cell division. So let's begin. So the first one is, I suppose we need to talk about is, one, why is cell division needed? And we start by thinking that we started life as a single cell. So the zygote that you were was formed from the fusion of egg and sperm. You were one single cell. To now, you are billions of cells, as the teenager that you are. And that process is entirely made possible because of cell division. So cell division occurs for a number of different reasons. So cell division can occur because of growth, repair, replacement, or reproduction. And there's 2 types of cell division in eukaryotic cells. So, like us, we're eukaryotic organisms, that we have to know. First off is mitosis, and mitosis is responsible for growth, repair, and replacement. Meiosis is responsible for reproduction. So mitosis is responsible for when we're growing, so from example from that zygote to the billions of cells you are now, cells replicating themselves and your body growing and needing to have more cells in them. Cells also die over time. So we need to replace them. And also when there's a damaged cell, we need to repair them. And that's all done through mitosis. Whereas meiosis is responsible for reproduction. Meiosis is responsible for producing eggs and sperm for the human race and for any eukaryotic organism. The first stage, no matter if you're looking at mitosis or meiosis, is that you have to go through DNA replication. So every cell in your body contains 46 chromosomes, 23 pairs. And every cell has an exact copy of the chromosomes that were found in the zygote that you once were, the one single cell that you once were. So every single cell in your body has every chromosome that was in that zygote. They're identical. Now, different sections of that chromosomes and the DNA may be active at different times, but in every cell of your body, there is exactly the same DNA. That means in order for that to happen, DNA must be able to copy itself to create copies so that there's a copy in every cell in your body. Now the steps that we do that is the first step, the double helix of DNA separates from each other, so think of like a zip being undone, and we'll go through this in class with the model. Step 2, the nucleotide bases are exposed, and then individual nucleotides that are floating around in the cytoplasm pair up with those exposed bases, and following the rules of complementary base pairing, I'll line up next to, for example, if there's an exposed T, and a free-floating A will come along and pair with that based on the complementary base pairing. The A goes with T, C goes with G. Step 3 is the sugar phosphate backbone will now be formed on the individual nucleotides, which makes a new strand of DNA. This process occurs for both the unzipped strands of DNA. So at the end of DNA replication, when we've had one molecule of DNA unzip itself, we end up with 2 molecules of DNA, new molecules of DNA. And we call that semi-conservative. So we can see that in this picture here, this is the idea of DNA replication happening. So the DNA molecule itself has unwound, and we call this the replication fork. The purple is obviously the old strand of DNA that's being unzipped, and the red strand is the new one that's being produced. So we can see from this one strand here, as we unzip it, we're actually creating 2. Now we call this the semi-conservative model because each of the new molecules of DNA is made up of one old strand of DNA and one new strand of DNA. And that's what semi-conservative means.